The recent discovery involving the enzyme ADAM19 and its role in gut inflammation and cellular aging has generated significant excitement in the scientific community. At first glance, the findings appear to offer a novel pathway to mitigate age-related gut disorders by controlling rather than eliminating senescent cells. However, when examined more critically, this study also highlights several unsettling truths about the biology of aging, the fragility of the gut barrier, and the complexity of manipulating inflammatory signals within the body. While the research suggests that targeting ADAM19 may reduce harmful inflammatory responses from senescent cells, the broader implications of this approach reveal a more precarious and uncertain road ahead, both for therapeutic development and for our understanding of the aging process itself. The study conducted by researchers at the Buck Institute for Research on Aging and published in Aging focused on how the metalloprotease ADAM19 affects the senescence-associated secretory phenotype, SESB, the group of inflammatory molecules emitted by cells that have entered senescence. These senescent cells, often thought of as zombie cells, no longer divide but persist in tissues, contributing to chronic inflammation, immune dysfunction, and tissue degradation. What this research confirms, disturbingly, is just how deeply ingrained and persistent these harmful cell behaviors are. As we age, the accumulation of senescent cells becomes inevitable, and with them comes a storm of inflammatory proteins that not only damage the gut lining, but also likely disrupt broader physiological systems. The implication is grim. Aging doesn't just slow the body down, it actively turns it against itself. While the researchers demonstrated that inhibiting ADAM19 in fruit flies, mice, and human cells reduce signs of gut damage and cellular aging, the study also exposes how interconnected and delicate the body's inflammatory machinery truly is. ADAM19, after all, is not an isolated actor. It is part of a larger family of enzymes that participate in immune signaling, tissue remodeling, and cellular communication. Any attempt to suppress it, even with promising outcomes, risks unforeseen consequences. One enzyme's inhibition may help reduce SASB proteins in one context, but could impair healing or normal immune responses in another. This raises serious concerns about off-target effects, especially if such treatments are proposed for chronic or preventive use in aging populations. Another layer of concern lies in the nature of xenomorphic therapy itself, the concept of modifying the behavior of senescent cells rather than eliminating them. While this approach may seem less invasive than senolytic strategies, which aim to destroy senescent cells, it may also be less effective in the long term. Senescent cells are not just passive emitters of SASB. They are metabolically active and highly resilient. Suppressing their inflammatory output may offer temporary relief, but the underlying damage they cause, both structurally and chemically, might persist or adapt. There's a real possibility that these cells, once silenced, could resume harmful activities or develop resistance to xenomorphic interventions. Worse yet, chronic suppression of key enzymes like ADAM19 could inadvertently promote the survival of damaged cells that should have been cleared, further burdening the tissue environment over time. Even more troubling is the central role the gut plays in systemic health. The gut barrier is not merely a digestive interface. It is a critical immune organ that influences everything from inflammation in the brain to autoimmune conditions and metabolic diseases. Any weakness or imbalance in gut function can rapidly spiral into widespread physiological disruption. The study shows that when ADAM19 is active, it helps cleave and release certain SCSB proteins at the cell surface, an action that appears to worsen inflammation and gut permeability. But modifying this pathway must be approached with extreme caution. It remains uncertain whether ADAM19 has essential functions that might be compromised by inhibition, especially in tissues undergoing rapid turnover or repair. The gut is a complex, self-regulating environment where even small perturbations in enzyme activity can result in cascading effects on microbial balance, immune surveillance, and nutrient absorption. Additionally, while animal models and in vitro human cells provide valuable insight, The translation of such findings to real-world human aging is notoriously difficult. Fruit flies and mice age differently than humans, and their cellular environments are far more controllable in a laboratory setting. Human aging is influenced by a combination of genetics, lifestyle, environmental toxins, diet, medications, and microbiome diversity, all factors that are difficult to replicate or control in controlled experiments. What works in a mouse may fail or even cause harm in a person, especially one with multiple chronic conditions, a compromised immune system, or already fragile gut health. Another looming issue is the pharmacological tool used in the study, batamostat, a matrix metalloprotease inhibitor that, despite its early promise, has largely fallen out of favor due to poor bioavailability and adverse effects in clinical trials. Repurposing it for long-term use in aging individuals could reintroduce the same toxicity issues. Moreover, even if new, more selective ADAM19 inhibitors are developed, they would need to undergo rigorous safety testing across various age groups, especially considering that aging individuals often have reduced liver and kidney function, increasing the risk of drug accumulation and adverse interactions. Lastly, the very fact that such an intervention is necessary, one that aims to tame the inflammatory chaos produced by our own aging cells, speaks to a deeper biological vulnerability. As we grow older, the body doesn't simply wear out in a mechanical sense. It begins to emit destructive internal signals, turning basic cellular functions into sources of chronic damage. This research reinforces a sobering realization. Aging is not just a passive decline, but an active, destabilizing process that we may only be beginning to understand. While the identification of ADAM19 as a potential target offers an intriguing opportunity, it also underscores just how complex and dangerous the aging process can become when we attempt to intervene at a molecular level. The pursuit of therapies that modulate senescence-related inflammation may yet lead to important breakthroughs, but the road ahead will be fraught with biological uncertainty, therapeutic risk, and the hard truth that aging is not a single enemy, but a network of deeply embedded processes that challenge every system in the human body.